Hi, everyone. My name is Ellie Andrews, for those of you I haven't met yet, and I am the Specialty Crops Advisor for Sonoma, Marin, and Napa counties with BC Cooperative Extension. So thank you so much for joining us today for this Soil Arthropod ID webinar. Today we're going to hear from Amanda Hodson, Professor of Soil Ecology and Pest Management at UC Davis, as well as Kelton Welch, Research Specialist and Taxonomist at the Ecdysis Institute. They're both entomologists with a ton of experience and expertise in this area, and I'm really grateful they're willing to share their knowledge with us on this topic. It's been a popular topic that's come up a lot with growers in our region that I've been talking to over the last year. So excited to get some experts in on this topic. We're going to start with a presentation from Amanda about soil arthropod biodiversity, general ID, and what they do. And then Kelton will walk us through how to identify Symphylans and their natural predators. And there's going to be time for questions and answers after each presentation, and plenty of time for discussion at the end as well. And also, I know many of you will probably want to know about Symphylans integrated pest management in more detail. So I pre-recorded about a half-hour presentation on my YouTube channel that you can watch whenever is convenient for you. I'm going to put the link in the chat towards the end of the webinar. So, you know, for further resources on IPM and what you can actually do about Symphylans in the field, different strategies you could choose from, that's a great place to look, as well as the UC IPM website, and I'll link that as well. Okay, so with that, I'm going to hand it over to Amanda. Take it away.
Okay, it's so lovely to meet you guys virtually, and it's exciting to be here in my office and get to share a little bit about one of my favorite things, which is soil biology. So let me share my screen. Presentation. Okay. So hopefully now you guys can see what this is my presentation. Please say something in the chat if you can't. Okay, great. You can see it. So this presentation is kind of what's there on your farm, what might you encounter, and what are they doing? And there's a lot of soil biodiversity. I'm primarily going to focus on things that are on the soil surface. And at the bottom, we have a picture on the far right of a Symphylan that I'm not really going to talk a whole lot about because Kelton's going to go into them in incredible detail. But there's definitely some things that can be problematic in biodiversity. Not all soil biodiversity is rainbows and sunshine, but a lot of them could be considered beneficial, like the ones in the other two pictures. The little purple columbella eats fungi and cycles nutrients, and a predatory mite is there in the middle eating another columbella that it could also eat Symphylans. So it's a little jungle down there, and there's good and bad and ugly, and it's just an all beautiful mess. So we'll get into that a little bit. So there's been a lot of research lately on human microbiomes and our gut microbiome, and how what we eat influences the communities of microbes and things inside of us. And that can influence everything from how well we digest food to our mood, to how sick we get. And they're realizing that the soil is kind of the same. Around roots, there is a microbiome of microorganisms, and the interactions between those microorganisms and the things we feed them can have all kinds of important downstream effects that we're just beginning to understand. So soil biology is literally the ground underneath us. We're standing on it. It's supporting all of these ecosystem services for us. It's interesting because all of these soil organisms just sort of self-exist for themselves. They aren't for us or against us. They're all living their lives. They're living their best lives, from the bacteria down here on the lower right, to this picture with the black background that has all these different mites and columbella, and perhaps a Symphylan or a proteran, I'm not sure, Kelton can tell us which. And then there's nematodes here in this picture in gray, and there's even viruses living in the soil that are killing all of these things and helping cycle their nutrients. So all of this stuff is there, and they're all going about their business. But one of the outcomes for that are things that affect us, like perhaps a disease outbreak, or perhaps just nutrients being cycled, or organic matter being decomposed and soil organic matter being formed. So in that way, the soil biology is very responsive to what we do. And it's very alive and dynamic. So what that means is that we can make management decisions that can be sort of processed by the soil biology, and then we'll have an outcome. So in a way, we could sort of have a conversation with our soil biology through management and monitoring, and kind of learning what's going on and what's best in our particular system. So people talk a lot about soil health, and last year in World Soil Day 2023, the slogan was "Soils where food begins," and this was sort of promoting soil health. And it used to be that people talked a lot more about soil quality, and soil health seems to have much more of an ecological acknowledgement that there's all these interactions between the soil organisms, and that's helping to create these outcomes like carbon and nutrient cycling. So whereas soil quality might just be kind of about agronomic functioning, this is your soil texture, these are the amounts of soil nutrients, soil health is much more process-based and interactive. So I like to think of soil health as this synergy of a system that is functioning ecologically well. It's cycling nutrients and carbon and all of those things, but it's also functioning well agronomically. So we're humans, it's from our perspective, whether we consider the soil healthy or not. So we want it to be functioning well for itself, but also sort of what we want, which is probably to grow some crops there in harmony with the native soil biology. So in this diagram on the right, we can see a lot of interactions going on. This is a more perhaps natural ecosystem where we have a tree with roots, but this could also be an orchard, or a farm with crops. So those roots might be fed on by plant parasitic nematodes, and those would be eaten by predatory nematodes and predatory mites. On the surface, those leaves might fall, or your inputs of other crop residues might fall, and they would be transformed and shredded by these little critters on the top right here, your isopods or roly-polies and earthworms and columbolans, and they're going to break down the leaf litter into forms that the bacteria and fungi can further digest, and that will fuel this whole other soil food web of nematodes that are eating the bacteria, other nematodes are eating the fungi, protists are eating the bacteria, other nematodes are eating those nematodes, and this kind of cycle goes on and on. But at each node and interaction of that soil food web, nutrients are being released, carbon is being stored in the soil biology itself, a lot of beneficial things are happening. And as those nutrients are sort of slowly released back into the plant, it's much more available to the plant than just a pulse of fertilizer, this kind of slow release of the soil biology. So in just a handful of soil, there might be 50 billion bacteria, 50 million protozoa, which are these sort of single-celled animals. There might be 100 million fungus, 10,000 nematodes, which are these little tiny microscopic worms that are one of my specialties and very dear to my heart. There might be 1,000 arthropods, and we'll talk more about what those are, and maybe 1 or 2 earthworms. And a lot of these things you won't be able to see, but today I'll talk about some of the ones that you can see, because these are the things that might be indicators of the rest. We can't always see the bacteria and fungi or the nematodes, but we might get an indicator of what's going on by what you can see on the soil surface. Okay, so we talked a little bit before in that picture about what the soil biology does, but the microorganisms like the bacteria and the fungi, they're the ones mineralizing most of the carbon and nitrogen. And as they die, their little mucus and dead bodies kind of glue soil aggregates together. And soil aggregates are really important because they assist with water infiltration and protecting soil carbon. The microorganisms might also detoxify, pesticides and chemicals. These microorganisms might also be symbiotic with a plant, like mycorrhizal fungi, or rhizobium in legumes that help plants fix nitrogen. Or they could just be endophytes that are living on the surface of roots and are helping plants in ways we don't really even understand yet. There's a lot of them that help plants resist disease, or they might be a disease themselves. So those are microorganisms. The larger organisms, comparatively larger, and I'll show you a scale in a second, they're doing things like shredding the leaf litter and surface litter, decomposing it. Their feces, their poop, will help stimulate the growth of bacteria and fungi that helps with further nutrient cycling. They might make burrows, which increases soil porosity. And they might also increase aggregate stability themselves, like earthworms make casts. So when we talk about soil critters, there is a huge range of body sizes. The microbes on the left here are under 100 micrometers. That's, a 1/10 of a millimeter. So they're very, very tiny. These include things like viruses, bacteria, fungi, and protists. The microfauna, literally like the little animals, are things in between that 100 micrometers and 1 millimeter. So they're almost, you could see them. These include tardigrades, which are these tiny little water bears, what they're called, and they can form these resistant cysts and survive for hundreds of years in the soil. And then when there's water, they kind of reanimate and bumble around and eat things that are smaller than them. Nematodes as well, they're about 1 millimeter on the big side. So if you had a tube of water that had nematodes in it and held it up to the light, they look kind of like fairy dust in the water. And there's good nematodes and bad nematodes. And there's obviously nematodes that are damaging to crops, but there's also a lot of good nematodes that cycle nutrients and also eat other nematodes. Then there's mesofauna, which are animals that are bigger than 1 millimeter, but probably smaller than 5 millimeters. And these include things like mites that we'll talk about in more detail, your Symphylans, your Collembolans. These are things that you can see maybe moving on the soil surface if you look closely. And definitely if they were in some alcohol, you could see them floating there. And you could see them in more detail with a good dissecting microscope. And then there's macrofauna, which is, the big things. They're still little compared to humans, but in terms of soil animals, macrofauna are things bigger than 5 millimeters. And this includes things like an earthworm or beetles that live on the soil surface, termites, ants, things like that. And today I'm talking mostly just about these two things because these are things you will be able to see on your farm. So how can you see them on your farm? Well, you might just see them on the soil surface, but one way that Ellie and I are going to look at in more detail is to use pitfall traps. And a pitfall trap is just a cup that's sunk into the ground that's level with the soil surface. And we can put a little bit of soapy water or non-toxic antifreeze at the bottom of that cup. And that's there to prevent the animals that fall in the cup from eating one another. Because then you might come back and just find one giant spider. It also preserves them so that then you can come back a week later and empty out that cup into a strainer and then empty that into some ethyl alcohol and then just go look through it and see what you see. And that will give you a lot of information about what is living in that place and what fell in there over the last week. So I'll go through a couple things that you might see. So you would probably see a lot of what we call arthropods. And if it's been a while since your last biology class, I'll remind you what an arthropod is. It is a very large phylum, and it includes insects, spiders, and even crustaceans. It means anything that has an exoskeleton made of chitin, which is this kind of hard-like material. So this is on the right, kind of what iNaturalist and Wikipedia pops up when you look at arthropods. It has things like horseshoe crabs, shrimp, spiders, barnacles are arthropods. I forgot about that. Centipedes, columbella, millipedes, and then our friends, the insects. Things that we would find on the soil surface include beetles, roly-polies, which actually are crustaceans. They're not bugs at all. They're one of the only crustaceans to make it onto land. A bunch of soil mites, columbella, which I'll go into in more detail, and then centipedes and millipedes. So millipedes and isopods are roly-polies on the bottom there. What they're doing, if you found a lot of them in your pitfall trap or just saw a lot of them around, they're shredding decomposing plant material into smaller pieces. And this is a good thing because it makes that food more accessible to smaller organisms. Especially bacteria needs the particles to be very small so that they can decompose it. Fungi are able to bridge space a little bit better and can decompose things that are a little more tough. But any animals that can break it into smaller pieces will increase the decomposition rate and the nutrients that are cycled. Don't confuse the millipede up there with a predatory centipede. So they're both probably a good sign of soil health and a healthy ecosystem, but they're doing different things. So the millipede at the top is cycling carbon, whereas the centipede on the bottom is predatory, and it's going to be helping you control your pests. And the difference between the two of them is really just the number of legs. Each segment of the millipede's body, it's divided into these little chunks. On the bottom right has one pair of legs, and there's probably multiple on the millipede at the top. They're also going to move very differently. A predatory centipede will be fast and it will bite. A millipede that eats leaves, it has no reason to go fast. It's kind of lumbering along. It's moving very slowly, and you can poke at it and probably pick it up. The worst it might do is release some sort of bad smell because it thinks you're going to eat it. Okay. The next thing you might see are a lot of beetles. And most of the beetles that you see are going to probably be in the family Carabidae, and they're ground beetles. These are predatory, and they can reduce pests in agricultural fields. So you might see some on the bottom, that's a carabid beetle. One kind of diagnostic feature with that is that they have these sort of three parts. We'll have like a abdomen, a thorax here that's sort of round, and then a head that has these chelicera. So it'll be a little obvious that they are a predator. You might see some tenebrionid, like darkling beetles, some stink bugs. And if you look at their mouth parts, it might be obvious that they aren't going to be killing anything with those mouth parts. They're just kind of like a black beetle. But these predatory ones have chelicera, and you can see on the top, this tiger beetle has some pretty impressive pincher-like fangs. Tiger beetles I've seen in a lot of farms around here in orchards, and they're this gorgeous iridescent green, and they run really fast. They have these big, beautiful eyes. So these kinds of carabids are a good sign, and you can promote them by reducing tillage or having strips of vegetation. I've heard of people doing like beetle banks and strips of grass. But again, you know, you can have your own conversation with the farm that you're involved with. You can plant some vegetation strips and put out a couple pitfall traps and compare it to another area where you didn't have the vegetation strips, and collect some data and see how many beetles you have. Another thing you'll find a lot of in your pitfall traps are soil mites. In this picture on the right, there's 200 species of mites, and this was from 1 square foot of forest litter. So there can be a ton of mites, and mites are very neglected scientifically. There just aren't enough people that study them, but we do know that they're enormously significant for nutrient cycling and also pest control. And even if you, you know, you can look at some of these mites and you might be able to tell some of them by eye, but you might need to have a microscope to check it out. A cheap dissecting scope will allow you to kind of tell some general differences between these mites. But obviously getting them down to like species level is very difficult even for specialists. And we usually send them out for DNA sequencing now as well. So soil mites, they're related to ticks, and there are some species that are in the soil that are kind of parasitic on birds and things like that. But most of them are going to be preying on other soil animals or decomposing things. So some of them will prey on nematodes, those colembala, springtails, or other mites and insect larvae. They might be preying on Symphylans, and other ones are grazers. So the one on the top is a predatory mite, and the one on the bottom is more of a grazer. And the way to tell about predatory mites, you can see in this picture on the top too, they sort of have these long raptorial front legs. So if you can, whereas this one on the bottom, it just kind of has these little tiny legs. So this one needs these long legs to be able to actually grab its prey. Whereas if you're eating leaves, you don't need them. So predatory mites, you can see this one on the bottom is just rocking out. It's killing that insect larva. They will eat anything the same size or smaller than them. And on the right here is a snout mite in the family Bedelidae. And it has that long snout, but you can still see that it has that front legs kind of moving forward so that they're ready to grab a prey. So you'll see lots and lots of mites in a sample. It's going to be very difficult to categorize them, even if you're looking on some sort of app like iNaturalist. But you can generally say, okay, those ones have their legs in the front. They're probably predatory. And check out this website as well, Chaos of Delight. They have great pictures, great information all about different soil biology and what they do, as well as good pictures of Symphylans and proturans and deplorans. The other mites that you would see a lot of are orbatid or box mites. And these are mostly fungivores and detritivores. Detritus is just kind of any leftover plant animal material that's on the surface. So they're just decomposers basically. And they're called box mites or turtle mites because they have this very heavy armor on their body. They're very heavy sclerotized. And this protects them from predation by those predatory mites. It's kind of a jungle down there. You would find more of these on a farm where there's a lot of surface residue, maybe a no-till system, and they're involved in carbon cycling. And people use them often as indicators of soil health. So box mites are probably a good sign for carbon cycling. Another thing you will definitely see are springtails. And these are some different families of springtails. And the reason they're called springtails is because they have this thing called a furcula, which is a little kind of lever that's usually curled up underneath their bodies. And when they get afraid, they release it and they fly through the air. So you might see them on the soil surface and say, oh, what a cute little springtail. And then it's gone. It just disappeared because it released the furcula and just flew away, flung itself out of harm's way. So that might be one way to kind of tell if you had something in a pitfall trap and you're wondering whether it's a Symphylan or a Columbella, it'd be to look for that furcula. There's some that live on the soil surface and have eyes, like that one on the top right, the Entomobriidae. And there's some that are little and white and blind and don't have eyes and live deeper in the soil profile, like Onychuridae. And then there's ones that are sort of in between. So these Columbella or springtails are very, very ancient relatives of insects. They were one of the first animals to come on land, and there's fossils of them from the Devonian about 400 million years ago. They're found everywhere, and they're considered a very successful arthropod lineage. There's like about 10,000 species. Most of them eat fungi, and they are thought to aid in nutrient cycling in the soil. And they are also, you know, eating things and pooping, and that's helping to form these aggregates within the soil that help promote soil structure. So the one on the top is a Entomobriid Columbella, and the one on the bottom is in the family Hypogastruidae, and it looks kind of like a little sack of potatoes. So they come in all different kind of shapes and sizes, but they have 6 legs like insects, but they're pretty soft bodied. And they're very small. They're gonna be about a millimeter. So that was all I had about these little soil critters. This is a picture of a predatory mite in the family Anistidae. But I would be happy to take any questions or just have a fun discussion about what kind of soil biology you guys are interested in.
Thanks, Amanda. What a cool presentation. Oh my gosh. Wow. Just amazing photos, you know, and so there's so many different functions of all of these different arthropods. I mean, there's just so much more going on than I realized. You know, I'm not an entomologist by any means. And we had a comment in the chat, you know, this presentation is awesome. I agree. Thank you, Joey. Does anyone else want to throw anything in the chat, or, you know, you can put something in the Q&A as well. This is a webinar format, so, you know, feel free, you can use the chat or Q&A and just let us know if you have any questions.
I mean, you could have a whole course
Yeah.
on soil biology and say we're gonna have, you know, 10 weeks of soil biology with a 3-hour laboratory every week. So
I would love that. I would attend.
yes, roly-polies are crustaceans. They're adorable crustaceans. So next time you see one, be like, congratulations, you're the only land crustacean.
I'm impressed by how cute so many of these arthropods are too, including.
Oh, the Columbella just look like anime characters. They're adorable.
It's true. Okay. Any questions? I have questions, but I'm gonna give folks one more minute before I jump in. Any questions from the audience, or Kelton, do you have any questions for Amanda? Okay. Well, I'll just jump in. So you mentioned having like strips of planted grasses and, you know, stuff that's not crops. I'm wondering like how often, if anyone in the audience has ever tried that, or, you know, would like to try that, or if you guys have questions about that practice. I think that's something that a lot of farmers I work with would be really interested in. And I love the idea of doing a pitfall trap, you know, maybe several pitfall traps in that kind of area versus nearby without them. So yeah, I guess I'm wondering, this is more for the audience. Has anyone tried that? or is interested in using that kind of like intercropping of natural spaces in between rows or maybe between fields or garden areas. I think that would be really cool, but okay. Ooh, Laura, okay, awesome. Yay, questions. Can you suggest methods of collection to find out what soil organisms are present? Okay, so yeah, I guess Amanda, if you could touch on that, maybe in a bit more depth around the pitfall traps and maybe any other methods you'd recommend.
Well, I mean, I know people are sending soil samples off to people that do commercial sequencing and try to infer functions about soil microbes themselves. So that's kind of one level. I work with a lot of farm advisors and farmers. My lab does diagnostics of nematodes as soil health indicators. So we kind of assess the nematode community and say, okay, here's what's going on in the soil food web. You have a lot of bacterial-based carbon cycling or a lot of fungal-based carbon cycling, or you have a huge pest problem, or you have great ecological structure and lots of awesome predatory nematodes that are probably suppressing your pests. So that's kind of like the nematode level. I don't think people really do like commercial mite or arthropod analysis, which is really kind of sad. So, and it's also difficult because everything is so site specific. Everyone's farm is different. So that's kind of why, in a way, I've been encouraging growers to kind of start that local conversation with their soil biology and see what do they like and see what they don't like and see maybe what is it helping. Do you see less pest problems when you plant some strip with alyssum or grass and have more crabid ground beetles? That's an interesting question.
Mind if I jump in a little bit, Amanda?
Yeah.
So my job, we just sample insects all over the country. So I've done a lot of different sampling techniques. The one we use the most is soil cores. We actually just use a soil core and pull the bugs out. I really hate doing soil cores because they take forever to sort through. They're really labor intensive. Pitfall traps are wonderful because they're really simple and they're easy, but they collect only bugs that move a lot. And so you don't get bugs that stand still. So you're going to miss pretty much all your mites, all your springtails, things like that. We like to do quadrats too. You just put a frame in the ground and then you use an aspirator to suck up the bugs and look at them under a microscope. One of the unfortunate things with soils is that all the interesting bugs are really, really tiny. And so, you kind of need a microscope to look at them. So if you don't have a microscope, you go find a place to look under a microscope and then you can collect them in any way you want. Really, just grab a lump of soil or something like that and put it under a microscope and you can see what's under there.
Yeah, and I'll just add real quick that if you're interested in microbes, which is a whole other can of worms, no pun intended, I'm so sorry. Different can of worms there, but if you want to know about microbes, PLFA, which stands for phospholipid fatty acid analysis, gives you a really nice big picture look at microbial community composition, but that's kind of a topic for another day probably. And we have, let's see, a great comment and question from Katie. Thank you, Katie. I heard something recently about strips of alyssum encouraging insect biodiversity. I've also heard tell about this. Curious if there are any specific crops to encourage arthropods. Great question.
Yeah, I mean, I was reading about these beetle banks. I mean, I think the beetles like some sort of ground cover. It seems like, centipedes and things like increased stuff on the soil surface. Maybe I don't know if that also promotes Symphylans, Ellie. So I think that might just be the increased organic matter itself. But alyssum is also nice because it'll give you those parasitoid wasps and things that'll come in and also control like aphids and other leaf feeding things.
Yeah, it's interesting, where living roots in the soil, having plants there, that that's good. And having organic matter on the soil surface, it sounds like that creates a beneficial environment for centipedes and probably other soil arthropods. And unfortunately, that is also the thing, just that, Symphylans likes as well. So yeah, I have questions there too around, how to sort of balance that out and think through that. I mean, there are some plant species that Symphylans for a lot more than others. So I think that could be a good thing to consider too.
And
Go ahead.
oh, and in that presentation too, under the crabid section, there's a link to an article that is like an e-organic article about ground beetles. And it talked a little bit about some literature of things that you could add, like grass strips and stuff. But that's not necessarily my expertise, but
Thanks.
there's a lot.
Go ahead. Sorry.
What I would say is, like, I wouldn't look for specific crops for specific bugs. I think the best thing that you can probably do is find out what sort of things are native in your area and grow a variety of them. If you want to grow them around your garden or around a field somewhere, grow a variety of those things, and that's probably the best way to get bugs out there. Because if you're trying to look for one specific bug, probably not the best way for you to get the ecosystem services you're looking for. Try to look for a way to make a diverse habitat so the system can be healthy.
Yeah,
That's
and
the
what's
advice
gonna
level.
work? What's gonna work in your farm, you know? And a lot of this research, even that's on the internet, has not been conducted in California with California cropping systems. I think we're all just gonna have to adapt and be our own scientists. You guys are gonna have to be your own scientists a little bit and conduct your own micro experiments to figure out what works with some guidelines. Like, yes, these little soil critters that are predatory on the soil surface, they like leaf litter, they like some sort of cover, they like that probably increased humidity, I would imagine.
Yeah, I really like that, experimenting on your own farm or garden, seeing what works best in your context, using plants that are native, like you mentioned, Kelton. Also, I just threw a link in the chat to the Chaos of Delight website you mentioned, Amanda, which has
Isn't it great?
best photos of arthropods. I'm so impressed. Yeah, and Kelton, I think you had actually mentioned this to me, to us a while ago too. So it sounds like maybe this is a website you both are familiar with, but yeah, so that's lovely resource. I think so. Let's see, 2:40-ish. I think unless there's any other questions about Amanda's presentation, we could move on to Kelton's if that sounds good with you guys.
Yep.
Okay, awesome. Thank you so much, Amanda. Feel free to stick around if you'd like to. Okay. Nice, there it is.
Sorry, my computer's lagging. It took me a while to get the mute button, unmute button there.
That's okay. No worries.
All right, let me share again. All right. Self unmute before sharing. All right. Thank you, Amanda. That was a great presentation. So now I'm going to talk a little bit more specifically about Symphylans, a little bit more about identifying them, how you can find them out in your field, and how you can know if you've got them. All right. Ellie, I hope you don't mind if I take a minute here and just kind of talk about the company I work for. I work for a nonprofit organization called Ecdysis Foundation. We promote regenerative agriculture. I have to click to use a different. We promote regenerative agriculture, which is, I imagine that this is in the crowd. I have to really describe that for, but just in case, regenerative agriculture is sort of a series of farmer-developed practices. These are practices actually farmers developed more than researchers. And they're geared mostly toward restoring the health and vitality of soils that have been depleted by decades of industrialized conventional agriculture. So we work out of a working regenerative farm called Blue Dasher Farm in a beautiful nowhere, South Dakota, which is where I am right now. I'm in the middle of a farm here. We do actually collect data from all across the country. And so we're trying to get this big picture of what's going on with all those different farming systems across our whole country. We've even been in Canada, as you can see with my dots up there. And we have our sampling takes the form of this called the 1,000 Farms Initiative. This is probably the biggest agricultural experiment that's ever been conducted. We're collecting data on 1,000 or more fields, pastures, orchards, vineyards, all sorts of places. We're currently sitting at about 850, so we're really close to our goal there. And when we collect data, we collect a lot of data. We do a lot of arthropod sampling, which is what I'm mostly involved in. But we do a lot of other stuff. We do soil testing, we do plant forage analysis, we do bird surveys, we do nutritional analysis on the food, and socioeconomic surveys on to get at sort of like the economics for the farmers as well as their health and well-being. We're trying to get this like holistic view of all the benefits of regenerative agriculture. And I'm the insect taxonomist or the arthropod taxonomist, and so I deal mostly with these arthropods. And I've been with Ecdysis for 5 years now, and in that time I think I've examined it's over 1 million insects so far. So I see all kinds of bugs, including Symphylans, which is what I'm going to be focusing on mostly today. Now, if I'm not mistaken, this Andy Murray, I think this is the name of the guy from the Chaos of Delight. May have been one of these other people. So I have a lot of their photos here as well. I have a large number of Symphylans in my collection, maybe like 50 to 100. I don't know. But most of my photos look kind of like this one. They're dead bug suspended in alcohol gel. So I kind of cribbed a bunch of images off the internet so you guys can see what they look like alive, what the bugs actually look like, what you'll see them out in your fields or in your garden if you pick up a clod of soil and look for them. Well, let's start from a basic question here. Let's just ask ourselves, what is a Symphylan? Well, a Symphylan is, it's a Symphylan, right? That may sound like I'm being a little bit flippant, but yes, I am. I'm being flippant there, but I'm also being truthful because Symphyllan is kind of like, it's kind of its own category of animal, right? So you may be familiar with this sort of a classification system that we use, the Linnaean taxonomy, where kingdom, phylum, class, order, family, genus, species. Symphyllan is actually way up there at the class level, so it's a really high level category, which means it's really a distinct kind of organism. It's up there at the same level as things like mammal, for example. Mammals is one class of organisms. Birds and insects, they're all classes, and Symphyllan is actually a category on that level. So it's kind of, when I say a Symphyllan is a Symphyllan, I'm saying it's actually its own thing. It's also, Symphylans are not like, they're not very familiar with people, and so there aren't like actual words for like different kinds of Symphylans, or there aren't like words for Symphyllan in general. We just have this weird scientific word, Symphyllan, which it means something like close together or together, something like that, which I don't even know why it's called that, to be honest. It's just this weird scientific word. We don't even know how to describe it with human speech, I suppose. Of course, just because they're all the same level of category doesn't mean they're all like equal. You have thousands of different species of mammals and birds. You have millions of species of insects. Symphylans, there's only, it's estimated there's about 200 species. So they're not as diverse as these other classes would be, and they're not very well known. The one we care about, the species for this talk mainly is just the garden Symphyllan. But most of what I say about the garden Symphyllan will actually apply to pretty much all the Symphylans we'll see. So I'm just going to talk about Symphylans and not necessarily about that specific one. Let's see. So where do the Symphylans fit in the tree of life here? They are arthropods. Amanda has already introduced that to us. Arthropod, that word just means jointed legs. Like arthro, it means joints. So like words like arthritis, for example, come from the same thing. And then pod, like tripod, is feet. So jointed legs. And of course, they're named after these jointed legs. Their legs are kind of segmented. You can see that on every kind of arthropod that I've shown here on this slide. They also tend to have a segmented body, so you can see the body is divided into parts all the way down. You can see that on pretty much every one of these as well. I've got all kinds of things here from the arthropods from around the phylum. Now, a Symphylan is an arthropod, but I want to make it clear that it's not an insect. There are a lot of similarities between Symphylans and insects. Here I've got a drawing of an insect and another one here of a Symphylan. And you can see the similarities, right? You see the segmented body on both of them. You see they have their jointed legs, fairly similar. They also have antennae. Not all arthropods have antennae, but insects and Symphylans both do. And that's a sensory organ. It's for touching things. It's for actually tasting and smelling things as well. But there's actually some really big differences between insects and Symphylans as well. You see, with an insect, you've got the body is divided into 3 parts here: head, thorax, and abdomen. And you'll note that the legs of an insect all grow on the thorax. And so it's got this long abdomen part that sticks out way beyond where the legs are. Compare that to the Symphylan, you see they do have a distinct head. The rest of the body is just a series of segments, and there's legs that grow all the way along the body, pretty much all the way to the back end here. And where insects have 6 legs, Symphylans have 24, which is 12 pairs. Although if you talk to some people, they'll say this pair, tiny pair in the front here doesn't count. So they only have 11 pairs. You know, you can count them however you want, I don't care. Insects also have a few other things that you won't find on the Symphylan. Insects usually have eyes. Not all of them do, but most of them do. And a lot of insects, I don't know if it's most of insects, but it's a lot of insects have wings. And obviously Symphylans don't. Symphylans are eyeless. Their antennae are the only thing they have to perceive their environment. So it's not an insect, okay? It's an arthropod. It's its own kind of thing. Now, for completeness sake, I'll also tell you it's not an arachnid. It's another kind of arthropod. Here's a spider. Spiders have their own whole suite of different anatomical features. They're similar to insects in some ways. They lack antennae. They have 8 legs. Obviously, a Symphylans is not an arachnid. You can look at that and see that, okay? It's not an insect. It's not an arachnid. It's not a crustacean. It's actually, there's another kind of, it's its own category, but it actually fits in with a few other categories of arthropods, and this group is called myriapods. And Amanda's already told us a little bit about some kinds of myriapods. That word myriapod is a myriad of legs. Myriad is a word that means 10,000. It's used poetically sometimes to mean just a big number. You may recognize it like in a biblical context, and like Revelation, for example, that John would call it, said there's myriads of angels. You recognize it from that context. Some of these are pretty familiar. Amanda already talked to you a little bit about centipedes and millipedes. I don't know if you'll have ever heard about a pauropod before. They're even less well known than Symphylans are, but they're also the kind of myriapod that would be most easy to confuse for a Symphylan, because they are tiny, they are pale, they don't have any pigmentation or color in their body, and they have fairly similar size, fairly similar leg numbers, things like that. I'll just tell you up front here that the way you can tell them apart pretty easily is the antennae. Like pauropods have these really short antennae, and they're branched, and so they look like, it kind of looks like it has antlers, like a deer. And Symphylans will have these long antennae that kind of stretch out in front of it like that. Let's say, I'm going to show you, go back to some drawings again, let you have some comparisons. One more time here, I'm going to compare a Symphylan to a centipede. And you can see that the anatomy is very, very similar between these. They have the distinct head, and they've got the body that's just a long trunk with a lot of segments and a lot of legs on it. And the antennae are long and everything like that. So you can see a lot of similarities between them. When you look at them in the field, though, it's usually pretty obvious which one's a centipede and which is a Symphylan, because centipede is probably about 3 or 4 times bigger than the Symphylan. And that's on the small scale for centipedes. The smallest centipede is probably twice the size of a typical Symphylan. One of the other important things that I want to point out about centipedes that distinguishes them from Symphylans is this right here. This is called a forcipule, or a poison claw, venom claw. This is what they use. They actually inject venom into their prey. They bite, and centipede bites, I'm told, can hurt. I've never been bit by a centipede. I don't intend to get bitten by one, but that's important for a lot of people because centipedes can bite, Symphylans cannot. Now, I'll point out, we say bite, it's not actually biting you, because this isn't its mouth. Like this is a claw that goes on it's more like a leg. It's more like a hand. Basically, it's using its little finger to stab you. So it's not technically biting, but whatever. So that's one of the key differences there between a centipede and the symphylin. I have a slide here that's just, it goes through a lot of details. I don't think I'm going to spend a lot of time talking about all the different things here, but I'll point out a few things from it. Up here at the top, I'm showing all the different kinds of myriapods here, and I'm showing them, this is kind of a size comparison, I suppose. This would be some of the common centipedes and millipedes are about this size relative to a symphylin, and a pauropod is a little bit smaller than that. Symphylin is probably about 2 millimeters long, maybe a little bit shorter than that, maybe even 1 millimeter long. Some of the differences you'll see, the centipede is the only one that has those venom claws because it's carnivorous. Millipedes are herbivorous or fungivorous, and then symphylins and pauropods have kind of varied habits. They're usually fungivores, detritivores, but as you guys probably know, are aware, your symphylins will also eat plants. They'll eat the roots of your vegetables and stuff. You also mentioned that like centipedes, they kind of move fast. Centipedes, they're really agile, they're really quick, their body's really maneuverable, they can move around really quickly on top of the soil or in the soil. They also have a really good burst speed, so if you startle them, they can jump really fast and go really quickly. Millipedes will not move very quickly, so that's a really easy way to tell a millipede from a symphylin. One of the easiest things, again, beyond size, symphylins are very small, but also they're always pale colored. And so if you look and you find something that's sort of brown, reddish brown, or has like gray or blue or beige color or something like that, it's probably not a symphylin. It's probably a millipede or a centipede. Okay. Let's see, what else can I tell you? I feel like I've forgotten some things. Oh yeah, well, millipedes, first of all, millipedes have 2 pairs of legs on each body segment. So they have a lot more legs than any other kind of myriapod. Symphylins have 11 or 12, okay? I'm going to go ahead and just put up a slide. Like with this webinar format, I don't think I can actually do like a quiz for you guys, but I'm going to go ahead and put a couple photos. Maybe what I'll do, I'll just give you a couple seconds and let you like think to yourself, what kind of myriapod is this? And then I'll give a little discussion after a couple seconds. So let's look at this one first. What kind of myriapod do you think this is? Go ahead and kind of think about it yourself. It's got a pale body. It's got long antennae. So that could be a symphylin, except that's a lot more than 12 pairs of legs, right? And what's this on its head, right behind its head? Looks like that's actually a claw. That's a forcipule. That's a venom fang. So this is a centipede. This is actually a soil centipede. This is kind of a centipede that a lot of people don't really know a lot about, but they're very common. This is the biggest group of centipedes. They're just always under the soil surface, so you rarely ever see them on the top. Very important predators in the system, and actually might be very important predators of symphylins. Let's go to the next one. What do you think this one is? Again, we've got a pale body, we've got long antennae. We don't see those venom claws here. We just see the little tiny leg there. This is a symphylin. This is the garden symphylin that we're looking at. And you can see he's got 11 pairs of legs plus that little tiny one. This is kind of what we'll be looking for out in the field if you're looking for damage in your crops. Let's try a third one here. This one is one that a lot of people have mistaken for symphylins in the past. It's pale colored. It can be very small, just a couple millimeters long. But you see, again, it doesn't have very long antennae. And you can see on those legs, the leg pairs look like they're in twos here, right? Double pairs. This is a millipede. This is a baby millipede specifically. It's a very, very young one. And you can see these when they're very small, it's very easy to pick up a clump of soil and you see it in there and you think, oh, it's a symphylin. But it's not a symphylin, it's a millipede. You'll see that it moves very slowly. It's got really teeny tiny legs and a whole bunch of them. So that's pretty easy to tell apart if you look really closely. But it's also, it's pretty easy to confuse it if it looks fairly similar. Okay, how about this one? And again, this one's dark colored, so it's clearly not a symphylin. It does not have very long antennae. You can't see its face, so you don't know if it's got venom claws. But again, the legs are in double pairs here. So this is another millipede. This one's an adult millipede. And this is a kind that you'll find pretty commonly out in the soil. So don't confuse that one. It's pretty easy. How about this last one here? This one might be a little bit tricky. Again, you see a pale colored bug. You see a lot of legs, but not two legs for each segment. You see long antennae. This one could be very easy to mistake for a Symphylan, but it's not a Symphylan. You can look right here. You barely see it. This is the venom claw right underneath its head. And in fact, you see its head has some like reddish color on there. That means it's got a hardened head capsule. That's indicative of centipede. You're not going to see that on a Symphylan. So this is a baby centipede. It's very easy to confuse them that way. So have a look at those and see. I'll give you this presentation. You can look at the images and compare them later. I also wanted to give you a chance to see what they look like when they're alive. And so I've embedded this video of YouTube. It's just going to show this little Symphylans walking around in the soil. And this will give you an idea of what you're going to see when you turn over a little clod of soil. You'll see it kind of pop out of the soil there, and they actually kind of have this sort of weird nervous energy to them. They're kind of moving around. His little antennae are bouncing around, touching everything he sees. They kind of move quickly. You can see they turn really easily. Their body's real wormy. They move around like that. He's touching things with his antennae like that because he's blind, right? That's the only way he's got to perceive his environment. And so it's kind of like, you imagine like a blind person tapping around with a stick, trying to see things around them. It's also probably tasting some things in the soil and even smelling things in the air as well as it goes. So look for something like that. If you see it moving around like that, a millipede's much more lethargic than this. But that's what it's going to look like when you see it out in the field. Let's move on. There's a couple other types of bugs. I'm going to go through these fairly quickly. There's a couple other bugs that you could confuse for a Symphylan. Amanda already talked to us about springtails and some of the cool things about them. Most springtails don't look very much like a Symphylan. Like they're colored, they might be round, they might have a very stiff body, something like that. But there are a few like onychurid like this kind. They have a sort of a flexible body, it's segmented, they're very pale. So it would be pretty easy to confuse that for a Symphylan. Except again, short antennae. And you're going to look, it's only got 6 legs. And you'll see this long body, this basically its butt is sticking out way past its legs. And so you know that this isn't a Symphylan because its legs would fall to the end. Another one that we talk about is Diplura. These are interesting bugs. We do find these a fair amount in the soils. And I think they're pretty easy to mistake for Symphylans because they have a very similar behavior with the antennae. They're also eyeless. They have the long antennae and they move around like this. Their body's kind of wormy and stuff like that. But again, they're insects or they're closely related to insects. So they only have 6 legs. And so again, you'll be able to tell that fairly easily if you know what to look for. There's 2 kinds of diplurans here. This one has the long tails that basically look like another pair of antennae out the back. And then you've got this other type that sometimes are called forceps tails. They have this pincer on the back, and that's actually used for catching prey. Now, I've never actually seen one of these catch a prey item, but I'm told they actually bend the tail up over their body like a scorpion and grab it with a pincer, which to me just makes no sense because they're under the ground, and how do they have the room to do that? But I don't know. They're supposed to be pretty good predators. Probably would be good predators of Symphylans as well. Probably the smaller end of that, because I don't think they're quite big enough to subdue an adult Symphylan. You also might find a lot of beetle larvae in the soil, and there are a lot of beetles whose larvae live in the soil. And again, they can look a lot like Symphylans. They have a long, skinny body with all the segments and stuff. But again, they'll have short antennae. They'll have 6 legs, like all the other things on this page. And they'll have a long body out here with legs on it. They also tend to have some coloration. So you'll see the head will have sort of this reddish brown color on it. Well, that's for hardening so that their jaws can be really strong and everything like that. I also put this one up here. I don't think you're going to confuse a pill bug for a Symphylan, but it's possible if you get like a baby pill bug, a really small one, a roly-poly, they can look like a Symphylan because they're segmented. They have a lot of legs. So it's potentially you could confuse them, but I don't think you would because they're quite big and bulky. And again, so I think it's okay. That's just for completeness sake. Wow, I'm going a little longer than I expected. Okay, let's go quickly. I've got one slide here, just some biological notes on the Symphylin, let you know a few things about it, tell a couple of stories about it. So what I've noticed with the Symphylin, we don't have really good literature on them. Like a lot of the work that was done was back in the 50s and 60s and 70s, and there hasn't been a lot of new stuff since then. And so what they found in the 60s and 70s was pretty simple, basic stuff that's been known for a while. We haven't had a lot of good updates since then. Let's talk about their ecology. I say they're omnivorous here. The garden Symphylin, for what you guys care about, I guess they're mostly herbivorous. Like they feed on the roots of your plants, and they'll feed there in the springtime. They like the soil to be moist and humid, so while it's in the springtime, they'll be up there where the plants are just growing, and they'll feed on those plant roots. And then as the summer comes along and the soil starts to dry out, they're going to go down further into the soil column. So they can go a foot down there, maybe even like a meter down into the soil column. I'm switching my measurement systems there, but they can go way down into the soil column at that. And once they're down there, they're probably just feeding on fungus, detritus. They may eat worms and eggs. I put that in there. I'm not entirely sure that anybody knows, to be honest. Their reproduction is a little bit weird. I want to tell this little story because it is weird. The male and the female never like eat each other. The male just kind of places a packet of sperm out in the environment somewhere, and the female sniffs it out with her antennae and picks it up and collects it herself. So, there's no real sense of romance between these creatures. They just kind of do their thing. You see their eggs, they look kind of like the classic sea mines, right? They're just ball with a bunch of studs all over them like that. They'll lay them under the soil. You probably won't see them unless you're pulling soil up and looking at it. And even if you do, you probably won't have to identify the egg because the female will be sitting there guarding, most likely. When they're first born, they don't have their full complement of legs. They first hatch with only 6 pairs of legs, and then they'll add new pairs each time they molt their exoskeleton. They add a new pair of legs. And I only put several months to adulthood here because I'm not sure. I've seen some people report that it only takes a month and a half or 2 months to get to adulthood. It may take 3 months. Some people have said, I'm not sure about that. It may be different habitats, different locations, stuff like that. But it's a fairly slow life cycle for an insect, for an arthropod. I call them insects. There goes my credibility. All right. Let's see. Okay, so that's all I want to talk about in the biology. Let's move on to natural enemies. I've got just a couple of slides to talk about, briefly introduce a few types of natural enemies. And on the point, again, we don't have a lot of good data on this because they live under the soil. So a lot of those surface predators have trouble, having any sort of impact on them because these guys are always hiding under the soil. But there are some things we do know. Like Amanda talked about predaceous mites. This is one particular one that I was able to find. It's been confirmed by a few studies done way back in like the 60s and 70s, particularly this one by Barry, who discovered that each one of these mites from this genus, Ecdysis, could eat 12 Symphylans in its lifetime, and they have 5 generations in the time it takes a Symphylin to complete one. So you can imagine how many Symphylans they could be eating. It's really amazing. He wasn't sure they were abundant enough in agricultural soils because they're mostly in the woods. But there are other kinds of predaceous mites that aren't this genus that will be found in your agricultural soils. So look for this. You look for a mite this one Pergamos is kind of on the bigger side. It's 1 millimeter, sometimes up to 2 millimeters large, which is huge for a mite, even though I've called it small here. They're sort of this reddish brown color, kind of teardrop shaped. And the males have this one pair of legs that are like claspers that they hold onto the female with. And this is kind of the size comparison that you'd expect. Here's that dead Symphylin I showed you earlier, and this is about the size they would be. So it'd be a pretty good meal for a mite. Another one that they've talked about in those studies is a specific centipede. This is called Lamictes. This guy Waterhouse actually found that this centipedes, they have a reputation for being really voracious. They'll just attack and eat anything. And he showed that in his study. They would sit there and eat Symphylans as long as you keep feeding them, they'd eat them. And he found they even had a tendency to overfeed. If they ate 9 of these Symphylans one sitting, they would, basically die from it. So centipedes they would love to eat like these Symphylans if they could. But there's still questions about whether they're abundant enough to be useful out in the field. Especially in like an agricultural setting, they don't like disturbed soil very much. If you've got good like stones and stuff you set around, they'll like to have those sort of hiding places and stuff like that. So to identify this particular kind of centipede, you're looking for a small centipede, maybe about an inch long. This is one of the smallest of centipedes out there. Only 15 leg pairs. That's the smallest number of leg pairs you find on a centipede. And if you want this specific genus, here's some other characters you can look at. But I'm not going to go through that details. Generally speaking, any small centipede is probably going to be a decent predator for the Symphylans. So have a look for those in your environment if you want. Here's a couple others, types of predators that have been mentioned in the literature. These are called rove beetles. You can tell a beetle because it has the first pair of wings is modified into these covers, these that kind of cover the back pair of wings. It looks like sort of a shell. But with rove beetles, the abdomen sticks out way past the shell. The shell is kind of stuck up here. But you can find those a lot in soils. They're very common predators. Some of them burrow under the soil and stuff like that. Ground beetles. Amanda already talked about ground beetles. I've seen them mentioned in the literature as predators of Symphylans. I've never really seen what specific species of ground beetles will eat Symphylans, which is a problem because this is an extremely diverse family. There's a lot of different varieties. They're often on the soil surface, like Amanda said, but this is one that burrows. So that one's probably a really good predator. Ants have been mentioned several times. Ants are kind of a can of worms because you don't know what, there's so many ants, they do so many different things, you don't know which ones are going to be useful. And then there's some fungi that can attack Symphylans as well. I couldn't find an image actually of a Symphylan that's been killed by this fungus, so you get to look at a caterpillar instead. So if you can see a Symphylan that looks something like this, it's been taken over by fungus, take a photo of it because there really aren't a lot of photos of that out there that I could find. And then I didn't find any evidence that these were ever mentioned in the literature as predators of Symphylans, but I think they could be. The soil centipede, it has to be. Like these are really good predators. They can actually burrow through the soil like an earthworm. They can make their own burrows and stuff. So they're really good predators in the soil. These are probably really important. And you tell them because they're extremely long with lots and lots of legs, pale colored except their head is red. I mentioned the forceps tails earlier. They're probably decent predators. And wolf spiders. Wolf spiders might be pretty good predators. They'll eat anything that they find. They are restricted to the surface of the soil, so they might not do a good job. But they might still eat Symphylans. Let's see. Yeah, I guess I don't have a conclusion slide, so I guess I'm just going to drop us off right there. Just acknowledgement, thankful to some people here. Thanks for having me over here. Tommy Finster is the one who got me hooked up with these guys to come give this presentation. Thanks to everybody at Ecdysis who's helped me with my work in the past. And here's a bibliography if anybody in the audience is a particular nerd and wants to read stuff. Thank you, everyone.
Thank you so much, Kelton. That was awesome. Very detailed, thorough. I loved it. More great photos, diagrams.
Sorry, I went over there a little bit.
Oh, no, that's fine. No worries. We're good. We have plenty of time. So now we'll do questions from the audience. So go ahead and put your questions in the chat or use the Q&A, and I'll give folks a little bit of time. I have some questions I wrote down too, but I'll hold those till later.
Apologies, Pat. I didn't have the chat open, so I didn't see that. I hope it wasn't too obnoxious for you to hear my voice like that.
Oh, it's okay. I think when you're a little bit closer to the mic, I think it's easier to hear. Yeah, that's great. And I think it got a little bit better after the first couple minutes. But there does seem to be like a little bit of an echo. I could hear you pretty well, but okay. So folks,
I'm all the way in South Dakota right now.
Oh, say it again.
It just probably doesn't help that I'm way over here in South Dakota right now.
Oh, yeah, you might have spotty service potentially. South Dakota. Wow.
Too.
Yeah. What's the weather like there right now as we wait for folks?
It's been extremely warm. It's been almost 60.
Wow.
That's warm in South Dakota.
Yeah, man. Okay, well, thanks for joining us from so far away. I don't see any questions, so I'm just gonna throw one of mine out there. I've been thinking about Symphylans a lot lately because it is an economically damaging pest for a lot of folks I work with, unfortunately. So we know that they appear in hotspots in the field. You know, they pop up as this sort of circle of death, which is an unfortunate phrase. It's what several growers have used to describe their symptoms in the field. It's plant stunting, and it's really patchy. It's sort of in these circles. So I'm wondering, do you have any ideas about how these hotspots start, or if there might be any predictors, when it comes to maybe like a low-lying spot in the field, maybe where water, soil, moisture pools, or maybe there's compaction. Do you have any ideas about like what might be a predictor for Symphylans population growth just based on what you know about their biology?
No.
Okay, I don't think we
You know,
know that yet. Like from a research perspective, I don't think we do, but I figured, while I had you, I'd ask. But it sounds like
no,
we don't actually know that yet.
it's a great question. It's one of those frustrating things. I think like in a lot of cases like that, the hotspots may be something that just appears completely random. Like a lot of soil bugs, they don't disperse very well, so they can't like they don't move. And so once they're in a location, they kind of stay there. So that explains why the hotspots grow, but where they start is very confusing. I'm not sure.
Hmm.
It could be that you just had some soil in your field and on your planter or something, and you went through and a clod happened to drop in one location, and that's where they started. It could be something silly like that. In general, I think it's probably more or less random. Just happens to be a population there, and they grow. It's unfortunate and not very helpful, to be honest.
No, it is helpful to know what we don't know. I mean, I wish we had the answer, but I just figured I'd run it by you just in case there was something out there I'd missed. But Katie has a great question and comment in the chat here. I'll just read it for everyone. So Katie says, I've heard of using crab meal to encourage fungal growth that will parasitize some phylums, and I do personally use this technique, but really have no way of knowing if it helps. Is there any research supporting this?
I mean, it sounds reasonable to me. Like you put some rotting meat out in the field, and fungus will grow on there. I would be a little bit skeptical that the kinds of fungus that grow on crab meat are also going to crab meal, you say. Oh, I missed that. I thought you said meat. Meal makes a little more sense, but yeah, I guess it could. I'm not sure. I think that like if you just put meal out there, you're more likely to encourage fungus that will just decay, dying stuff that's already dead will just take decaying matter. I
I
don't
mean,
know
I've
that
heard of this like chitinaceae. There's like chitin degrading fungi. Um, and this is a thing. People sell pest control products that you add chitin, and then the idea is that it'll stimulate these microbes that will then attack the chitin of the living soil organisms. And I think there is some research in support of it, but again, super hard to know. I would imagine that Symphylans and other soil arthropods have techniques to defend themselves from fungi that eat chitin. So it's hard to know. It probably depends. I think there's probably very, very little research done on Symphylans, which is why we need to do trials or our own experiments.
yeah.
Sorry to say we don't know, but we really don't know.
Yeah, and then Katie just followed up and said, especially since they can move, right?
They
So
can move, yeah.
run away if they so choose.
Run away!
Yeah, and they're so vertically mobile. I'd imagine they could just retreat down, to lower levels of the soil in some cases. Yeah. Did you have any other thoughts on that, Kelton?
I mean, I think like the basic idea there is sound, like find a way to promote fungus and stuff. I mean, my answer would always be to find a way to make your system more like nature, make it a diverse ecosystem, put more stuff in there, try a bunch of different things. One of the problems with Symphylans is that they break the rules in that regard. They like that sort of habitat. They do really well there. But I suspect that the more diversity you can get into your field in terms of fungus, in terms of predators and natural enemies, probably going to be better off in that environment, in that sort of situation, than in a situation where you're growing one vegetable on bare soil. Yeah, that would be my suggestion: is try a variety of different things instead of trying to focus on one specific tactic that you can use to do one specific function.
Yes.
That's easy to do in a garden or a small scale, not necessarily in a commercial farmer. So in that case, you may have to try out things like this might work for you really well. I don't know. It's interesting. I'd love to see the results that you get from something like that.
Yeah, and I'm happy to connect folks after this event too, Katie, if you want to try like a small little trial and do like control versus crab meal. I mean, that's something we could set up pretty easily if you'd like. And we can share results with Kelton and Amanda. But another thing I was going to mention, Kelton, you just said, instead of relying on just one thing, try lots of things. That is something I talk a lot about, and lots of folks in Extension talk a lot about within IPM. So integrated pest management is a toolbox approach where you combine multiple strategies, don't rely on just one thing, and it's going to look really different. Like he was saying, depending on your context, even between gardens and between different small farms, different people come up with different, combinations of strategies that work best in their situation. So just bear that in mind. There's no one size fits all, but there's a lot of strategies at your disposal. And actually, on that note, this is a good time for me to put the link in the chat to a follow-up video on Symphylans integrated pest management that you can watch another time whenever it's convenient for you. So that goes into more detail about strategies. But do we have any other questions for Kelton about his presentation or anything related to Symphylans? I'll give folks another minute or two to throw things in the chat or the Q&A.
I just say, like, it's really nice that you guys have people like Ellie and Amanda, and they're in California, that are able to support this sort of thing, because like I said, the literature on Symphylans kind of disappeared after the 70s or something. And you see every IPM guide out there is citing studies from the 70s and nothing since then. So there's very little. So it's good that you have university affiliated folks that are working on this to support you guys.
Thanks, Kelton. Yeah, I get the sense that this pest is so difficult to study since it does show up in such a patchy way throughout the field. And I can, especially because I'm not an entomologist, but it's hard for me to imagine how to set up a randomized replicated trial with really consistent pest populations in all of, across the whole trial, because it is so patchy. So I could see how that would make it harder to study, compared to something else that's a bit more predictable or simple. But
yeah. Big issue being that they're under the soil most of the time. Can't see them down there. Can't get our pesticides down there. We can't get our predators down there unless they're already there. So disturb your soil and it's over for you. There's nothing soils take a while to regenerate after you disturb them too much.
Yeah, that's true. Okay, well, I don't see any other questions. I'll just ask one more that I had written down. I know a really common question I get about Symphylans from my growers here is whether they could be imported to your site in compost. And it seems like that's pretty likely. I know I've talked with Jim Leap about this. He's a retired UC Santa Cruz farm manager who's worked on Symphylans for decades and has been very supportive and open with his experiences, sharing information about this pest. But he said, yeah, totally. I mean, it seems possible, especially they might be able to survive like on the edges of compost piles where it's not quite as hot. So I was wondering if either of you, Kelton and Amanda, have thoughts on how can growers like test or ensure that their compost that they're bringing onto their farm or garden doesn't have some violence in it? Do you have any suggestions? I mean, maybe like a bait test with a potato or something, or just asking the person you're buying your compost from, hopefully they'll be able to tell you with confidence. But I don't know, do you have suggestions about how folks can just ensure they're not actually like bringing some violence onto their site in compost?
I mean, at least in California, there's pretty, I mean, I guess I would say try to go through like a reputable compost dealer and not just pick the cheapest one. And a lot of them will provide some sort of certificate that it's free of disease. I mean, I think they might have submitted it for some plant diseases, but maybe ask for that. And then, yeah, it's hard because probably once it's as soon as it comes to your site, they're going to say, well, it could have been, yours. It could be something that's there. I could see Symphylans colonizing compost as soon as it hits the ground because it's amazing and they want to live there. So I'm wondering that's where this is coming from, because an actual commercial legitimate compost operation should have their compost getting very hot in the center, to a degree where it wouldn't be great for a Symphylan to live in. But I think as soon as that compost comes to a farm and it's like in a pile and it's cooler, then Symphylans that are there are going to jump into that compost. So you could look, but I mean, by the time you're looking at the compost or baiting it or sampling it, it's already probably at your farm. So don't take compost from your neighbor, maybe.
Yeah, especially if they've had a Symphylan problem already.
Yeah.
I would kind of agree with you on that, Amanda. I think that what I saw was that some of the studies I saw were that Symphylans don't do so well at really high temperatures. They try to avoid that. They like kind of medium warmth. And so, yeah, in the middle of the compost pile at least would be pretty problematic for them. But, you know, if you're talking about pulling the compost off the top layers or something, you might get Symphylans in that. I think you might be more likely to get them even just like soil if you're doing potting soil or something like that. Um, so yeah, I guess you just. I never thought about testing it. That's kind of that's a frustration, isn't it? Trying to test a soil, you have to put it out in a pot or something and check it before you use a bait trap, like Ellie suggested. Do that before you put it out there. I guess would be the best thing you could do. But that's kind of annoying.
Yeah, it seems like the, lowest time input option is just to get, compost from a really reputable source where it's getting up to temperature, consistently throughout the pile, getting up to temperature as best as reasonably possible. Yeah, it's good to know that they prefer more moderate temperatures. I kind of got that sense, but it's reassuring to hear that from you that, if your compost gets up to temp, that should hopefully take care of it, for the most part, at least. Yeah, fingers crossed. Again, more research. We need more research on this. Okay, any other questions or comments, anecdotes? Feel free to put them in the chat.
I'm gonna have to take off. It was
All
lovely
right.
to meet with you guys.
Thank you so much, Amanda. We really appreciate you being here. Thank you.
Yeah, email me anytime.
Okay,
Bye.
that's good. Bye. All right, well, I'm not seeing any other questions or comments, so I think we'll wrap up. And thank you again, Kelton. We really appreciate your expertise in this. This was a super cool presentation, really informative. And yeah, this is being recorded, so I'll go ahead and share it with anyone who was unable to attend but interested in watching. So thank you so much for your time, and I hope everyone has a great afternoon.